All right, now we're going to go over paper 6, which is the alternative to the practical for biology. This is IGCSE Cambridge International Examination, CIE, from May-June 2016, and this is variant 1, so 0610/61. All right, let's get started.
Now, shall we begin?
Question 1. Metabolic reactions in cells produce toxic chemicals which can be converted to harmless or less toxic chemicals. Hydrogen peroxide is broken down using the enzyme catalase, which is found in most cells. Figure 1.1 shows this reaction. Okay, we have hydrogen peroxide broken down using catalase into water and oxygen. A student investigated the effect of alcohol, ethanol, on the activity of catalase found in potato using 3 pieces of potato cut to the same size. Figure 1.2 shows these pieces of potato. Okay, we have 3 rectangles of potato. A1. Measure the length, width, and height of one of these pieces of potato and record your results in table 1.1. All right. So this is table 1.1. You take out your ruler, measure the length, the width, and the height. Okay, and it has to be in millimeters because it says so in the headings. I measured 30 millimeters, 10 millimeters, and 10 millimeters. Okay, if you printed it out on a different size piece of paper, you might get something different. As long as you know how to use your ruler, that should be okay. You can use the dimensions that are in the mark scheme, or you can use the dimensions that you measured. That's okay. This is just your own practice. Step 1. The student labeled 6 test tubes, 1, 2, 3, 4, 5, and 6, and used a syringe to add 10 centimeters cubed of hydrogen peroxide solution to each of the test tubes. They cut potato piece A to obtain 2 slices of similar size. So they took piece A, this rectangle, and said, that looks about right, I'll cut it in half. All right, so they have 2 pieces of potato called potato A1 and A2. The student placed the free end of a delivery tube into a large test tube containing water. They placed one of the slices of potato piece A into the hydrogen peroxide solution in test tube 1. The student immediately placed the rubber bung attached to the delivery tube into test tube 1 and pushed it in as tightly as possible as shown in figure 1.3. So this is the setup that was being described. Okay, in this test tube over on the right hand side, we have water, and the delivery tube is in the water so that you can see any bubbles coming out. In the test tube on the left hand side, we have hydrogen peroxide, and we have A1, we have the small piece of potato piece 1 inside the hydrogen peroxide, and as the catalase on the outside of the potato reacts with the hydrogen peroxide, or causes the reaction to occur, then bubbles of oxygen gas will bubble through. Because remember, the 2 products are oxygen and water. Water will obviously just mix with the hydrogen peroxide. Step 6. They counted the number of bubbles released from the delivery tube in 3 minutes. Step 7. The student repeated steps 4 to 6 for the second slice of potato, piece A, using test tube 2. So they repeated it with piece A with the second piece. Then step 8, they repeated steps 2 to 7 for piece B using test tube 3 and 4. So they did B1 and B2, doing the whole process for 3 minutes. And step 9, they repeated steps 2 to 7 for piece C using test tubes 5 and 6. The students used a tally to count the number of bubbles. So this is the tally of their results. So we have potato piece A1, A2, B1, B2, C1, C2, and they just tallied them. You're going to have to convert those tallies into real numbers. 2. Prepare a table to record the student's results. Your table should show the number of bubbles produced by each slice of potato in 3 minutes and the mean number of bubbles produced by each piece of potato A, B, and C. Complete your table using the results from figure 1.4. All right, so whenever you do a table, the left-hand column should always be the independent variable. That's the thing you change. So this can be the potato. All right. The next columns are the things you're measuring. So first of all, you're measuring the number of bubbles in 3 minutes. And the final column is the mean number of bubbles in 3 minutes. The way that I always do this, because you want to try to make this as neat as possible, is I always fill in the first column without any lines in. You can do it a different way if you like, if you find that useful. And I do this so that I can draw on the lines using a ruler, and then I know that everything in the columns next to it will line up to it. Okay, so this is potato, I'm going to say potato A1, A2, B1, B2, C1, and C2. All right, now I'm going to fill in the lines using a ruler. Okay, so this is my table that I've drawn in. Keep in mind there are other ways of doing this table correctly. There's generally more than one way to fill in a table, to create a table correctly, but you have to have all the information in the headings. You have to make sure your units are in the headings, and we have to make sure that your independent variable is in the first column and everything else is after that. Okay. So now we have to record the data that was recorded. So this is 5, this is 18, this is 12, this is 3, this is 11, and this is 10. Okay, let's record this. So now let's record the means. Okay, so if we do 5 plus 3 divided by 2, we get 4. If we do 18 plus 11 divided by 2, we get 14.5. And if we get 12 plus 10 divided by 2, we get 11. Now, a mean number of bubbles, you don't get a half a bubble. So you may not want to write down 14.5, you can write down 15. So you write down 15 instead of 14.5 because you don't get half bubbles. B1. Suggest why the free end of the delivery tube was placed in water before adding the potato slice to the hydrogen peroxide solution and connecting the test tube to the bung of the delivery tube. Well, the reason why is because you need to try and collect as much of the oxygen as you can, and if you don't have it already set up, you might lose some of the oxygen at the beginning of the experiment. So to try and collect all of the oxygen as soon as the experiment begins. All right. 2. Explain why the bung of the delivery tube must fit tightly into the test tube. Well, that's so that you don't lose the oxygen out the sides of it. So it's to prevent oxygen leaking out so results will be comparable. You could also say it's to create accurate results or consistent results or reliable results, valid results. Also, making sure that the bung is tightly fit into the test tube means that pressure can build up in the test tube so that bubbles will go out the other side. C. The pieces of potato that the student used in their investigation were soaked in different concentrations of alcohol for 24 hours. Potato piece A was soaked in 20% alcohol, B was in 2% alcohol, and C was in 10% alcohol. Suggest the relationship between the number of bubbles and the activity of the catalase. Okay, so we have to look at our results. So the most bubbles were produced in test tube B, and the least bubbles were produced in test tube A. Test tube B had 2% alcohol, and test tube A had 20% alcohol. C was in the middle, and that was at 10% alcohol. So it looks like the less alcohol that the potato pieces were soaked in, the more bubbles. If there's more bubbles, then catalase is working better, is doing more reactions. So the lower percentage alcohol soaked up by the potato, the more bubbles were produced, so the greater catalase activity. And 2, compare the activity of catalase in the potatoes pieces A, B, and C. And this is a very similar question to the first one, but it's not the same. This one you have to be specific. A, you're just saying lower percentages good for catalase activity. B, 2, you have to say about potato B having the most catalase activity and potato A having the least. So potato B has the most catalase activity and potato A has the least. You don't have to mention potato C if you say the most and the least, because if one has the most, one has the least, then the other one has the middle. 3. Predict the number of bubbles that would be produced in 3 minutes if a piece of potato was soaked in 50% alcohol before being placed in hydrogen peroxide solution. So you know that at 20% alcohol, then it was 4 bubbles produced. So you have to guess how many bubbles are produced if there's 50% alcohol. Okay, so it is a guess, but you know that if it's 50% alcohol, it reduces the catalase activity. So anywhere between 0 and 4 is okay. You could say it'll be the same as at 20% alcohol, or you could say it would be less. So you could say there'd be less than 4. You could say no bubbles. You can say 4 bubbles even. So it has to be 4 or less. D1. State one variable that has been controlled in the student's investigation. Describe how this variable was controlled. So you choose a variable and you have to say how that particular variable was controlled. Okay, so one of the things that was controlled is the time that they were measuring the bubbles for. So the time for measuring bubbles. And this was for 3 minutes. So they counted for 3 minutes for each piece of potato. Okay, other things you could put down. You could say that hydrogen peroxide volume was controlled, the volume of hydrogen peroxide, or the concentration of hydrogen peroxide. You can't say the amount because amount could be volume or concentration. And how did they control it? They controlled it by measuring out 10 centimeters cubed of the same strength solution. You could say that the potato size or the length or the volume or the surface area was controlled, and that was by measuring the same dimensions for each piece. You could say the type of potato was controlled because the pieces were cut from the same potato. And you could say that the time of soaking in alcohol was controlled because they soaked each piece for 24 hours. So any of those would be good, but only do one. 2. The method of measuring the oxygen gas produced is a source of error. State one reason why this method is a source of error. So basically they just counted the number of bubbles. Now some bubbles can be little and some bubbles can be large. So small bubbles will have a different volume of oxygen to large bubbles. So bubbles are different sizes, so they are different volumes. How can you control this? Well, you can measure the volume of bubbles produced, the volume of oxygen produced, using a gas syringe. So you can measure the volume of gas using a gas syringe. 3. Identify the source of error in step 2 and state why this is a source of error. Okay, so step 2 is when they cut the pieces of potato into 2 slices of a similar size. Okay, so the reason why that was a source of error is because you don't exactly know if they were exactly the same size. They eyeballed it. They just guessed to see if it was the same size. So the volume of the slices of potato may not be equal, or you could say the size of the slice of potato, the surface area. There's a few different things you can put down, but they may not be equal. And the reason why that's a problem is because when you get different sizes, you also get a different surface area. And the catalase that you're studying, that you're looking at, is on the surface. So the surface area is different, so each piece will have different amounts of available catalase. 4. Describe a control experiment that the student could carry out for this investigation. So remember, the control experiment is basically the situation that's very similar to the other situations where you expect nothing to happen. And if something does happen, you need to go back to the drawing board. But at least you have something to compare it with. So a suitable control experiment for this experiment would be to do the same experiment, but do it in water. So soak 1 potato in water. But this is a 2-mark question. You need to say something else. You're describing the experiment. So basically you want to say that you're doing everything else exactly the same. Use the same procedure as the original method. And 5, predict the results expected from the control experiment described in 4. So if you soaked 1 piece of potato in water, you'd expect there to be lots of bubbles because soaking it in alcohol slowed down the number of bubbles produced. You would expect more bubbles of gas produced than any other trial. E. State one safety precaution required when ethanol is used in an investigation. So it doesn't have to be this investigation. It can be used in any investigation. Well, ethanol is extremely flammable. So one thing you want to do is make sure you keep it away from flames. Keep ethanol away from flames. If you have to heat it, do it in a water bath. Don't do it over a flame. Other things you could say, the generic wear gloves or goggles or a lab coat, they also work with ethanol, even though it's not really dangerous if you get ethanol spilt on your skin. It is in your eyes, it's bad on your eyes. On your clothes, it's not going to do much for most of the time, but yeah, those are general safety things. Use tongs, but again, it's not really going to do much. The most dangerous part about ethanol is generally keep it away from flames or wear goggles because you don't want it in your eyes. F. In an investigation into the effects of alcohol on the nervous system, people were asked to carry out a test on their reaction time. The person being tested looked at a colored block on a computer screen. As soon as the color changed, they pressed a button. The time taken to press the button was recorded by the computer, and this was their reaction time. 20 people were tested before and after consuming a drink containing the same concentration of alcohol. Table 1.2 shows the results of this investigation. All right, so it doesn't all show up on the screen, but basically they are not in order. It's just they tested person 1 before and after consuming the alcohol. Okay, and there were 20 trials. 1. Calculate the mean for the reaction time after consuming alcohol. Write your answer in table 1.2. Okay, so the mean for before is already written in for you. The mean for after is, where you fill it in. Now, they don't say leave a space for your working out because that's kind of irritating, writing it all out and then having to type it into the calculator. It's just another place you can go wrong. So just basically you type in 225 plus 316 plus 286 plus 261 plus 340, etc., etc., etc. for the 20 trials, and you divide it by 20. And the answer you will get is 280. There. 2. Plot a bar chart to show the mean reaction time of the people tested before and after consuming alcohol. Okay, remember a bar chart, you have bars, but they do not touch. They're not related to each other. They're not actually, it's not a continuous set of data. So you want 2 bars on this, and you don't want them to touch. So if you look at this, I'll just do it at the top. You probably want 1 bar going that set of blocks, and another bar going that set of blocks. Okay? Okay, so first of all, we're going to label the axes. Now on the x-axis, you don't have to label the whole axis, you just have to label underneath the bars. So 1 bar is before drinking alcohol, and 1 bar is after drinking alcohol. Okay, you want the label as close as you can to being under the bars as possible. And so the y-axis is the mean reaction time in milliseconds. So mean reaction time in milliseconds. Now let's look to see what you have to go up to. So you only put 2 numbers down. One of the numbers is 240 and the other one is 280. So as I said, it doesn't all show up on the screen. So there's 7 large squares. A reasonable number to go up by is 50. So we say 0, 50, 100, 150, 200, 250, and 300. You can even go up to 350, although you're not using that. So you're going between 200 and 280, really. So just under 300, 240 and 280. So looking at this, we have 50, we're going up by 50. We have 10 squares per large square. So that means 50 divided by 10. So each small square is 5 milliseconds per square. Okay, that's just so you know how to work it out. You don't need to write that on the side. So the first one is 240. Okay, so I drew a line at 240. All right, so that is the first bar. You don't need to color it in because you have the label underneath. You want to draw it in a sharp pencil, but you don't want to draw really hard just in case you make a mistake and you need to erase it. You do want to draw it hard enough though so that the examiner can see, but you should just do it in pencil. So don't worry about coloring it in. You'll waste too much time. And that's the second line drawn in. That's all you have to do. You do not want the bars touching. You want them nice, clear bars. 3. The range of reaction times recorded before consuming alcohol is 180 to 310 milliseconds. 180 to 310. Use table 1.2 to identify the range of reaction times recorded after consuming alcohol. So what we want to do is we want to look through these numbers and look for the smallest number and look for the largest number. So if you look through this 220, that is the smallest number. And 350 is the largest number. Okay, the other results are down here, and those are the 2 numbers. So it's 220 and 350. That's the range. That's all you're looking for, the largest number and the smallest number. So the range is 220 to 350, and it even has milliseconds already written down for you, so you don't have to worry about that. Now we can move on to question 2. Question 2, the drawing question. Figure 2.1 is a photograph of a cross-section of a vascular bundle in a leaf. Line A to B shows the length of the vascular bundle. All right, this is the vascular bundle. We have line A to B. We know we have a xylem vessel here, and so these are xylem vessels. This whole section, though, is xylem tissue. So these other smaller ones are also xylem. Okay, and this is a phloem tube. All right. A1, make a large drawing to show the different regions of the vascular bundle shown in figure 2.1. Do not draw any individual cells. Identify and label on your drawing the position of the xylem vessel as shown in figure 2.1. Okay, so when it says don't draw any cells, you have to be very clear. This is a cell. That is not tissue on its own. This is one cell. All you are doing are drawing the positions of the major tissues. And you can see that because they're different colors. So you can go all the way around the outside. Better than that. You probably want to have one going about like this, one going here, and put one here. So those are the only things you draw. You do not draw the individual cells. This is called a plan diagram. But remember, when you're doing drawings in biology, you have to do one smooth straight line, one smooth continuous line, not straight, not with a ruler, certainly not in this one, but other times you never use a ruler in a biology drawing except for labeling. And you shouldn't be able to tell where you started and ended. So if you do a circle like that, you'd lose a mark. Don't sketch the lines. Don't do coloring in or shading. I mean, you shouldn't show that this is darker by coloring it in or by putting stipples, little marks to show texture. Don't do any sort of hatching at all. Okay? And you want to do everything in as close to the same proportions as the original. But remember, the examiner knows you're not an art student, so there's a little bit of leeway. It doesn't have to be 100% perfect, but it has to really look like you tried to draw exactly what was in the picture. Okay, that's a reasonable drawing. It's not perfect. It's pretty good. It shows the different tissues reasonably well. You can sort of see this line right here, so you have to be careful about those. Remember, when you do drawings in biology, you need to use a sharp HB pencil. Very sharp though. And don't draw really hard, draw lightly, so when you erase it, you can't see what you just erased. And this says, identify and label on your drawing the position of the xylem vessel, as shown in figure 2.1. So the xylem vessel was here. Okay, but that is not actually, you haven't drawn in one xylem vessel, you've drawn in, that's where the xylem is. So you could just say it's xylem. All right, so that's the xylem. That's all that you need for this diagram. So where do you get the marks for this drawing? Well, one of the marks is that you've drawn an outline, that all the lines are one single, clear, and unbroken line. You can't tell the beginning and end. That the size should be at least half of the space you're given. Not going over any of the writing, but at least half the space you're given. The detail, you need to make sure it's the oval shape that it should be. You've drawn in the phloem, as it says, and you need one other area. So this is the phloem and another area here, and then two other areas. So the xylem and the area around it. And the label, you need to make sure you have one line. It has to be a line with a ruler, not an arrowhead, because an arrowhead is wrong. That's not how you do drawings in biology. And obviously it has to be to the right spot, and it has to be labeled xylem. Okay, and that's all you get marks for. So don't worry about having it 100% absolutely every lump and bump. Okay, and if you spend time drawing cells, A, you'll lose a mark for drawing cells, and B, you'll waste a lot of time. So read it carefully. Don't draw cells if you don't need to. And remember, a xylem vessel is a cell. 2. Measure the length of line AB as shown on figure 2.1 and include the unit. So measure on this diagram here the length of A to B using your ruler. And when you do that, you could easily get a value of 58 millimeters. If you're doing it off of the actual exam, you'll get 58 millimeters if you know how to use your ruler. If you're doing it off of a piece of paper that was printed off onto a different size, you may get a different number, but that's okay. As long as you know how to use your ruler, that's all right. Now, when you go into your exam, make sure that your ruler is actually a good quality ruler you can actually read the markings for. All right, so 58 millimeters. Does it matter if you measure it in centimeters? No, doesn't matter at all, but you do have to make sure you've included the units. So this is 58 millimeters. So mark on your drawing a line in the same position as A to B. Okay, so the line in the same position goes about like that. And don't forget to do the beginning and ending of the line. And it says to measure it as well. So when I measured this, I get 115 millimeters. Don't write it over here, write it down below. So measure the line you have drawn, and the length of line on drawing is 115 millimeters. So the magnification equals length of line on drawing divided by the length of AB. Calculate the magnification of your drawing using the information above and your answers, and show your work. So magnification equals the drawing over the image, which is 115 millimeters over 58 millimeters. And if you divide those, you get 1.98, no units because that crosses out. Now 1.98, you don't know it's 1.98. You might be, if you measured a slightly different angle, you might have a slightly different amount. You never do it too precise. You always round it. So this is, you could say it's times 2. You could say if it was 2.4 or 2.5 or 2.6, you could say you could do a decimal there. But if it's say 2.1, you could still say times 2 or 2.1. Either of those would be okay. So you don't want to make this number overly precise. This is not a precise measurement. 3. State one way visible in 2.1 in which the xylem vessel is different from the phloem tube. So there's a few things you could say about this. You could say that the xylem vessels have a larger area because clearly they do have a larger area than the phloem tubes. You could say that the xylem vessel walls are actually thicker than the phloem vessel walls. You can see that in the picture quite clearly. You can also see that the xylem vessels are rounder than the phloem, although that's a little bit more ambiguous because it's a bit hard to see the phloem very well. Xylem vessels are definitely quite round. So you can do any of those 3, but you only do one. So you can say xylem vessels have a larger cross-section area than phloem tubes. All right. B. The walls of xylem vessels are supported by a chemical called lignin, which can be stained by a red dye. This makes the xylem vessel walls easily seen when using a microscope. Use this information to plan how you could find the position of the vascular bundles in a stem. Okay, you want to know where are the vascular bundles in a stem? First of all, you have to find a stem. You could say cut a stem from a flower. It doesn't have to be a flower, just cut a stem. But you're designing an experiment, so you want to say some specific details. So cut a stem from a flower. Then what do you do with that stem? You put it in red dye. So you put the stem in red dye. You can also, instead of put the stem in red dye, you could put red dye onto the cut surface of the stem. That's all right as well. Then you want to let the stem sit for a certain amount of time so that the dye can be absorbed into the stem, let's say 10 minutes. All right, so that's just an arbitrary number I've chosen. It should definitely have absorbed some by 10 minutes, but it depends how long the stem is really. Then what do you do there? After that, you cut off sections of the stem. So once you've cut off sections of the stem, then you can look at the stem and see that the red stained parts are the xylem. So starting from the beginning, you cut a stem from a flower, you put the stem in red dye, you let the stem sit for 10 minutes to make sure that some dye has been absorbed, you cut sections of the stem, then the red stained xylem will indicate the positions of the vascular bundles. And that's it. That's the end of this exam. I hope you have found this useful, and if you have, please press the like button, and we'd really appreciate it if you'd subscribe. If you have anything you'd like to say to us, any questions, any comments, we'd love to hear from you. So please write them in the discussion section below, and have a great day.